Well, thank you, Ken, and thank you all, and thanks especially to Matt for all the hard work you've done in getting the grant and getting us all together here. This has been a really interesting symposium, and I hope what I have to say actually ties together a lot of things we've already heard from a variety of different speakers. I'm going to focus on aflatoxins, and for good reason, because aflatoxins are very potent chemical carcinogens. They're a natural product. They're produced by a mold, Aspergillus flavus, that grows on corn and peanuts. Everybody thinks about peanuts, but in fact the majority of exposures, probably worldwide and certainly in the United States, is actually from corn. The FDA regulates aflatoxins very carefully. The level of allowable aflatoxin in corn and peanut products in the United States is 20 parts per billion. And keep that number in mind because it sounds very small, but you'll see in a moment that it may not be that small. We know a lot about the disposition of aflatoxin and its mechanisms of carcinogenesis. It's activated to an epoxide that binds to DNA and causes mutations, particularly RAS proto-oncogene activation and inactivation of p53. And we know from lots of epidemiology studies that aflatoxin is a very important contributor to the global incidence of liver cancer worldwide, especially in regions, or almost exclusively in regions, in which hepatitis B virus is endemic. Now, I became very interested in aflatoxin decades ago, not just because it's a really potent carcinogen, but because there's some really interesting and dramatic species differences in susceptibility to the carcinogenic effects. In the first study that was done in laboratory animals, 15 parts per billion of diet in the diet to rats gave almost 100% incidence of cancer. Interestingly, 150,000 parts per billion in the diet of mice gave no liver cancer. So as toxicologists that rely on animal models to predict human response, this is a bit of a conundrum. Are humans like rats, or are they like mice? And you would come to very different conclusions based simply on those data. I'm also interested, and we'll talk a fair amount about the role that we might, that diet and chemicals, in a good way, chemopreventive agents, might in fact be able to modify susceptibility to aflatoxin carcinogenesis, and I'll show you some work on that. But in order to understand that, we really need to understand a little bit about the mechanisms for this species difference in susceptibility, and of course it's related to biotransformation, at least the species difference between rats and mice, and the big question is, how is this relevant to humans? So here are the data just to show you a second study that was done by Jerry Wogan a long time ago. Around 100 parts per million in this study here gave essentially 100% incidence of liver cancer, 50 parts per billion giving 80%. And here's the FDA tolerance level for aflatoxin in the human diet. So we're not talking about extrapolating orders of magnitude here when you talk about potential exposures. So the biotransformation of aflatoxin is important. This is the structure of aflatoxin, this difuranol coumarin compound, and this double bond here is critical for its metabolism. It can be detoxified through P450s, through hydroxylation at different points on the molecule. But what's really important is the formation of this epoxide, and work done in my lab and Fred Gingerich's lab over the years have demonstrated that cytochrome P450, 1A2, and 3A4 are responsible for forming aflatoxin epoxide in human liver. And work in our lab suggested that CYP1A2 under normal conditions is far more important, even though there's less 1A2 in the liver. The Km is very low for CYP1A2 with aflatoxin, and it's much higher for CYP3A4, although CYP3A4 can certainly do this. CYP3A4 actually makes aflatoxin Q1 as well, with a ratio of about 10-to-1 detoxification to activation. Interestingly, CYP1A1, inducible, that we've heard about, forms aflatoxin M1 exclusively. It doesn't form any epoxide, but CYP1A2 forms both epoxide and a little bit of M1. So the metabolism is a little complicated, and there are big species differences in terms of which P450s do what to the molecule. Interesting, glutathionetransferases, of course, are a target, this is a substrate for glutathione transferase detoxification, diol formation through epoxide hydrolase, we showed in a study is probably also important, especially in the absence of much glutathione-transferase activity. And ultimately, what dose of aflatoxin causes DNA addicts that lead to liver cancer depends very much on understanding the ratio of these different metabolic pathways. Decades ago, when we started this work, we did a very simple study, which was to look at cytosolic activity of glutathione transferase towards aflatoxin epoxide in different species, and what jumped out at us right away was total cytosolic activity of mouse was gangbusters. It really had a lot of activity towards aflatoxin epoxide. Rats had a fair amount, monkeys had about tenfold less than rats, and humans had essentially no measurable activity, which was kind of scary because one would hope that it might be the other way around. We then went into looking at which GSTs were responsible for this, and we identified one and cloned this gene probably 30 years ago for mouse GST A3. And this is the form that has incredible activity towards aflatoxin epoxide. Rat GST A3, which is 86% homologous to this, has a very low, that got flipped around, a 0.3 should be over there, very low activity towards aflatoxin, even though it has some measurable. But interestingly, there's a form of glutathione-transferase in the rat called A5 that is actually the ortholog of A3 structurally. It's about 93% similar to this, and it has relatively high catalytic activity. Human alpha-class GSTs that we find in the liver have no measurable activity towards aflatoxin, even though I have lots of activity towards other substrates, such as CDNB. And interestingly, human glutathione transferase M1, which is polymorphic in the human population, has very little activity, but Fred Gingerich did find some measurable activity, as we have as well. And keep this in mind for something I'll point out in a little bit. Now, so this clearly suggested that expression of the mouse GST A3 in their liver was what was protecting them from carcinogenesis. And we proposed this decades ago. Finally, somebody made the knockout mouse for MGST A3. We tried and weren't successful. But Elek et al. Published a paper a couple years ago that in fact demonstrates that what we found is in fact really true. These are DNA addicts in the wild-type mouse expressing MGST-A3 and the knockout mouse, and you'll see about a 300-fold difference in DNA addicts from this. So this is pretty conclusive evidence that the reason that mice don't get cancer from aflatoxin is because they constitutively express GST-A3 in their liver. Now, interestingly, the GST-A5 form that I mentioned in rats that has considerable aflatoxin activity is inducible. Its expression is regulated through the antioxidant response element and the NRF2, keep one NRF2 pathway. And we showed, as Tom Kensler did as well, that the rat GST-A5 is in fact inducible by ARE type of activating pathways. There's a lot of information in the literature and epidemiologic literature suggesting that diets high in cruciferous vegetables are associated with reduced risk of a variety of types of cancer. And why that's the case, of course, has been a big question. One area of emphasis for a long time has been on these so-called glucosinolates that give rise after digestion in the gut to the release of certain isothiocyanates. And one particular glucosinolate called glucoraphanin, which is present in broccoli and broccoli sprouts, gives rise exclusively to the glucosinolate called sulforaphane. So sulforaphane is this structure here. Paul Talalay at Hopkins first explored the ability of sulforaphane to modulate chemical carcinogenesis and induce biotransformation pathways. Interestingly, broccoli sprouts contain high levels of sulforaphane, and if you go to your local grocery store and you see broccoli sprouts and you buy it, if you look carefully on the label, it'll say patented Johns Hopkins University. And this because that strain happens to have a lot of sulforaphane in it. Now, sulforaphane has been shown by Tom Kensler and others to be a very effective activator of the NRF2 ARE pathway. And lots of efforts have been focused on understanding the implications of that. You can go into the health food store and buy broccoli sprout extracts and even sulforaphane extracts. And many years ago, when this story first started, we actually fed freeze-dried broccoli to rats and then looked at aflatoxin DNA addicts after a couple of weeks, and it had a profound effect. It greatly lowered the DNA addicts. And the reason is the GST A5 level that has aflatoxin activity is not normally constitutively expressed in rat liver, even though the gene is there, but it's inducible. So the question is, could we do that in humans? And we've heard already a lot about the NRF2 pathway, antioxidant response element. I'm not going to go into this other than to say that there are a lot of genes regulated by this pathway, including, at least in rodents, many glutathione So we were interested in this question: can diet modify the biotransformation of aflatoxin in human liver in a way that's positive? John Grootman and Tom Kensler did a variety of really elegant studies, both in animal models in rats, in which they demonstrated that agonists or activators of the NRF2 pathway were very effective in inducing GST A5 in the rat and lowering DNA addicts and even lowering preneoplastic foci in tumors. They then subsequently did some human studies with a drug called Ultipraz in China, which is a good activator of the NRF2 pathway. And basically, they didn't find a great deal of evidence of an effect, but they did see some reduction in aflatoxin DNA addicts in the urine that they were using as a biomarker in this population. Subsequent studies showed that the interpretation of that is somewhat complicated because Ultipraz happens to be a very effective competitive inhibitor of cytochrome P451A2. So whether the protection was because you were lowering activation or inducing GSTs was not clear. It turns out that sulforaphane doesn't have that effect on P450s. It seems only in rodents to activate the ARE pathway. We were skeptical that this would work in humans. And the reason that we were skeptical is, first of all, I showed you that the alpha-class GSTs in humans don't have any activity towards aflatoxin. So inducing a gene that doesn't have any activity isn't going to do you any good. And secondly, we looked and looked and looked and could find no evidence of an antioxidant response element in the GST genes, in any alpha-class GST gene. And studies in human hepatocytes also, we found no induction. And so we decided that we would put this hypothesis to the test in human hepatocytes, do a very simple experiment, take human hepatocytes, pre-expose them to sulforaphane, give them a dose of radiolabeled aflatoxin, and look at DNA addicts, and also do a gene expression array analysis to see what happens. And I was pretty sure that what would happen is when we gave sulforaphane to these hepatocytes that it wouldn't do anything to aflatoxin. So this was basically the experimental design that I just talked about. And I was wrong. We did this experiment with a couple of different isothiocyanates, sulforaphane and phenethyl isothiocyanate. And at the higher dose of 50 micromolar sulforaphane, basically DNA addicts were at the level of detection. It completely protected. And I was surprised at that. I was interested in the mechanism of that, of course. We showed that if you gave this at the same time that you gave the aflatoxin, you didn't pre-treat, there was no protective effect. So it required something prolonged, like induction and changes in gene expression in order for this effect to occur. So we did the typical array analysis. In those days, it was the old-fashioned way of a 20,000 gene array. And we looked at a lot of different genes in 10 and 50 micromolar sulforaphane and a few other NRF2 activating agents. And we did some different hepatocyte preparations and looked at the vehicle control. And to make a long story short, what we found was that no GSTs were induced, as we had sort of expected. But what happened in these hepatocytes was that CYP3A4 messenger RNA disappeared. Sulforaphane shut down the expression of GST, or of CYP3A4, sorry. Now, I mentioned earlier that CYP1A2 is important in activating aflatoxin, and that's absolutely true in vivo. But when you isolate hepatocytes, and those of you that have done this know this, P450s go down to almost nothing. And then over the couple days of culture, they start coming back up. And what happens is 3A4 comes up really fast, 1A2 really comes up slow. So in these experimental conditions, 3A4 is the principal activator of aflatoxin under these conditions. And so the lack of DNA adducts that we see following exposure of human hepatocytes to sulforaphane was not due to induction of GST, any GST, but rather due to, apparently, to inhibition of activation via blocking the expression of CYP3A4. Now, I'm going to follow up on that story, but I wanted to come back to one last thing. Remember when I showed you that GST-M1 had very, very little activity, but some measurable activity in human liver, and human alpha-class GSTs have no activity. 50% of you sitting in the room are homozygous null for the GST-M1 gene. You don't have a gene. And we started looking at this, and we had done about 10 or 12 human livers. So we went back and genotyped these, just looking to see. And in the absence of any treatment, just looking at genotype in human livers, there is a threefold difference in AFB DNA adducts. Those that are GST-M1 null having 3 times higher adducts. So this suggests that that remarkably little activity that we had in the absence of anything else is actually very important. And in fact, a number of epidemiology studies in China have suggested that those with GST-M3 null genotype are at increased risk for liver cancer from aflatoxin. So it goes to show that even a small amount of activity can actually be very important. Now, interestingly, when we treated with sulforaphane, the level of adducts, of course, was lower because it knocked down CYP3A4. But in fact, there was no additional protection through the GST-M1 pathway, so it didn't appear that GST-M1 was induced under these conditions either. So we know in humans that 1A2 and 3A4 are important in activating aflatoxin, that CYP3A4 expression was greatly downregulated by sulforaphane. And that sounds like, wow, maybe we could treat people with broccoli and get rid of their aflatoxin through inhibiting CYP3A4. But you all know that that is not without consequences. 3A4 is the major P450 metabolizing drugs, and that would not be a good thing to knock down your CYP3A4. Talked about that, the lack of activity in humans, that mu class could be important, and that polymorphisms in these pathways might be important. So looking back at the dietary approaches pathway, there are some problems here if you're focusing on inhibition of P450s rather than induction of GSTs. Now, the question of how does sulforaphane lower CYP3A4 expression caught our attention because that's very important. We looked, I wasn't a P450 regulator guy, but my colleague Ken Thummel down the hall was, and he had a new postdoc, Cheng-Cheng Zhao, who had joined his lab. So we put Cheng-Cheng on this question to say, you know, how is sulforaphane lowering expression of CYP3A4 in human mice, or in humans, and as well as humanized PXR mice, which I'll talk about in a moment. And our initial hypothesis was that sulforaphane was somehow interfering with the pregnane X receptor or CAR. We didn't know which. We knew both of them were important in driving 3A4, and our first look was at PXR. We've heard a little bit about PXR already, pregnane X receptor, and the fact that it's a ligand-activated nuclear transcription factor with endogenous ligands that can upregulate genes, including especially CYP3A4. And the question then was basically, could sulforaphane somehow block this effect? And it's very important because, again, PXR is involved in the regulation of a lot of different, expression of a lot of different biotransformation pathways. Further, there are a lot of drugs that are ligands for PXR. And this is a major cause, one of the major causes of adverse drug-drug interactions is if you take a PXR ligand, it upregulates 3A4, and all of the other drugs that you're taking at that time get metabolized much faster, and they don't work. So if there was an effective antagonist to PXR ligand activation, that could actually be pharmacologically very useful. And there are a lot of examples of drug-drug interactions that are mediated through ligands activating PXR. So we did an experiment in human hepatocytes, said, gee, I wonder if we could block PXR activation with sulforaphane. And there's two different ligands here, rifampicin, which is a very potent ligand for PXR, and RU486, which is also an effective ligand, but less potent and effective than rifampicin. And basically what you'll see here is if we get rifampicin to human hepatocytes, we get a whopping induction of CYP3A4, which we knew we would. And in the presence of 10 or 25 micromolar sulforaphane, that induction at 25 micromolar is completely blocked. And we see the same kind of a protective effect with rifampicin, a different ligand. And if you look at midazolam metabolism, which is mediated only by CYP3A4, again, we get protection of the rifampicin upregulation. And we thought that was pretty cool because there were no known and are no known effective non-toxic antagonists to the PXR receptor. And if one could develop one, you might be able to co-administer it with drugs like rifampicin and block the induction of CYP3A4. And the reason that that's clinically important is tuberculosis is the most common opportunistic infection in HIV/AIDS patients. HIV/AIDS patients worldwide now get first-generation antiretroviral drugs, which are great substrates for CYP3A4. If you get tuberculosis and you're taking an antiretroviral, you can't use rifampicin because you will make your antiretroviral therapy ineffective. And so this was a clinically very interesting idea that we might be able to co-formulate rifampicin or co-formulate sulforaphane with rifampicin and protect the induction. And so we actually got some money from NIH to do a clinical trial, a small clinical trial on this idea. And it was a 3-arm study in which we took broccoli sprout extract rather than sulforaphane because we would have to go through INDs and all of these things. But since it's a natural product and you do an extract, you can do that. It simplified the protocol quite a lot. And people were their own controls. We did it 3 times. They got sulforaphane for 7 days, or they got rifampicin for 7 days, or they got rifampicin plus sulforaphane for 7 days. And on day 1 of each one of those legs of the protocol, they were given a small dose of midazolam, and we did a pharmacokinetic clearance assay of midazolam, which is a good measure of phenotype of CYP3A4. Then at the end of the 7 days, we repeated the midazolam assay. And for this to work, then what we were looking for was we expected the AUC to go way down with rifampicin treatment because CYP3A4 activity goes up. We didn't expect to see anything with sulforaphane on its own. Maybe an increase in the AUC if it was blocking endogenous ligands. But the combination of the two, we would expect to see the AUC not change in the presence of rifampicin, which would have been the clinical desired outcome. And we looked at a number of other things, such as urine and stuff. So this is basically the design over a 3-week trial. These are the data, and I'm going to show them in a way that's more understandable, but these are different pharmacokinetic parameters. You can look at clearance or AUC or the terminal slope of elimination. But basically, this is what we found, which is that in the treatment with rifampicin, you can see that the AUC went way down, like we expected. Okay, so this is showing that rifampicin in this study did what we wanted it to do. It induced CYP3A4. The AUC for rifampicin went way down. Broccoli sprouts didn't do anything. Unfortunately, broccoli sprouts really didn't do anything because in the leg that included rifampicin and sulforaphane together, or the broccoli sprout extract, we got essentially the same level of reduction with rifampicin. So this didn't work in the clinical trial. Exactly why, I don't know. But we did notice that if you took the people that had lots of CYP3A4 activity, the top 75% of midazolam clearance, and you looked at sulforaphane, the AUC actually statistically went up, suggesting that in people who have endogenous ligands that are driving their expression of CYP3A4, that we did get some inhibition of endogenous ligand driving CYP3A4 expression. And the conclusion from all of this is that it's probably a pharmacokinetic effect rifampicin is such a potent, strong ligand that we just couldn't get the dose delivered to the liver over a long enough period of time in vivo, even though, of course, in hepatocytes you can do that because it's right there. Now, just very quickly, at the time we started this experiment, there's also a species difference in the pregnane X receptor such that rifampicin is not a ligand for mouse and rat PXR, but it is a ligand for human. When she and others developed humanized PXR mice, and we tried this experiment in which we did isolated hepatocytes from humanized PXR mice, and we found exactly what we found in human hepatocytes. We got perfect protection of sulforaphane against rifampicin induction. We then did our little mini clinical trial in humanized PXR mice and found the same thing we found in humans, which is it doesn't work in vivo. So while that was disappointing, it was an interesting proof of principle of the value of humanized mice in predicting human response, because here we got the same in vitro response and the same in vivo response that we found in the clinical trial. Those are basically those data there. So I'm going to skip over that and just spend the last couple of minutes talking about a new study that is not ours, but it's really quite fascinating, and it follows on the work that I've been talking about. And this was a study that Tom Kensler and Bill Roebuck, who you've heard me talk about before and doing a lot of the sulforaphane work in the past. Michael Sporn discovered another activator of their NRF2 pathway called CDDO-IM, and it has a name about this long, so nobody even tries to talk about what its actual name is. But this compound is an incredibly effective activator of NRF2, keep1 NRF2 ARE pathway, and all the genes that we talk about that are regulated by the ARE go way up with this compound. And so they thought, well, let's do a real study of cancer chemoprevention with this really potent ARE agonist in rats. And so they took rats and did basically a 2-year study. They did a short-term study as well, looking at pre-neoplastic foci in rats that were given 4 weeks dose of 200 micrograms per kilogram per day of aflatoxin, which they knew would cause lots and lots of tumors in these animals out a year. And then they treated with or without the CDDO-IM. And basically what they found, and this is the pre-neoplastic foci using GSTP, what they found was the foci, of course, after 28 days on aflatoxin treatment, 13% of the volume of the liver was as GST-positive foci. That's a lot of tumor, pre-neoplastic tumors for 28 days of treatment. And that CDDO-IM basically completely eliminated the foci. Well, that was amazing, but does that hold for tumors? Well, in fact, here's their survival curve out for 100 weeks of age. And what you will see is these are the animals in the control group that did not get CDDO, just aflatoxin, 100% liver cancer mortality after 100 weeks. And you will see the CDDO-IM goes on out, and there were no tumors in those animals. So they got 100% protection of aflatoxin carcinogenesis in these animals. Now, if the mechanism of that protection were solely the upregulation of GST A5 in these rats, so they didn't get any aflatoxin DNA addicts, that would make a lot of sense. But what they found was that, in fact, it did do this. It lowered DNA addicts quite a lot. This is the FAPI addict, which is a ring-open furanidopyrimidine addict after aflatoxin binds to N7 guanine. It depurinates and this opens, and this is a stable addict that's thought to be mutagenic. And what's really interesting is while they got a substantial reduction in addicts, both of the N7 guanine addict as well as the FAPI addict, they didn't get zero. There were a lot of addicts in these animals in the treated group, but they didn't get cancer. And this was argued in the paper and argued by a couple of editorials that were commentaries. I was asked to write a commentary on this, and I focused on how this is or isn't relevant to humans in terms of the mechanism. And Ken Olden wrote a commentary about the observation of this the lack of concordance between reduction in DNA addicts and reduction of tumors, suggesting that in fact there might be thresholds for carcinogenesis, so the implications there are quite profound. Although they didn't do a dose response, and these doses were very high, and so you couldn't really argue about a linear nonlinear type of response. It clearly was a remarkable demonstration of the potential for chemoprevention of cancers, even in the face of a very potent carcinogen. My own guess is that CDDO-IM is doing something in addition to upregulating GST-A5, while it's doing that, is doing something else downstream that is very important in the carcinogenic process that keeps these animals, even though they've got adducts, from developing tumors. This paper just came out a couple months ago, and I thought it was very interesting. Thank you for your time. I'd like to thank some of the people in my lab that did the work. Emma Jane Polton was a graduate student who did the clinical trial and put up with everything that you have to put up with doing clinical trials. Cheng Zhao was an amazing postdoc that did discovered the mechanism of sulforaphane as a PXR antagonist in about 6 weeks. It's probably the only time I've ever had somebody that comes in every day with perfect data, and I started worrying that he was making it up. It was so good, but he was amazing, and a lot of other collaborators, including Steve Strom, who until recently was at Pittsburgh and provided us with hepatocytes, and Tom Kensler and Bill Roebuck for sharing their slides with me, and Frank Gonzalez for our humanized PXR mice. And when we got some mice from Wenji early on in this process too, and we ended up using Frank's for our study, but Wen was one of the early discoverers of, or generators of the PXR null as well as the humanized PXR. That'll stop. Thanks.
Thank you very much, Dave. Great presentation. We have time maybe for a couple of questions for Dave. Gary?
Yeah. Getting back to eggs, one of the reasons we were intrigued about this model is the known sensitivity of turkeys to aflatoxin. And in the comparator study that we've done of different carcinogens, non-carcinogens, which is in press in toxicological sciences, aflatoxin B1 was by far the most potent genotoxin in the eggs. The thing I want to ask you about, though, suppose as a non-genotoxic comparator, aflatoxin B2, and interestingly enough, it was also genotoxic.
Interesting.
And we've been able to find one paper by Jerry Wogan where he reported that duck liver homogenase convert B2 to B1.
Yeah, there is some reduction. There can be some reduction with aflatoxin, with a dehydrogenase of some sort. And Jerry reported that many years ago. So also B2 preparations, you have to be very careful that they're completely free of any B1 contamination because it's so potent. The turkeys are actually interesting. They don't have a glutathione just like humans. So they don't detoxify aflatoxin well at all, and so they're actually a better model for humans perhaps than mice in that regard. And Roger Coulomb has shown that. He's also shown the P450s that activate it in turkey. So I'm not surprised that you got a robust response in the turkey.
Nathan, last question.
Yeah, so Tom's original assumption was that the broccoli sprouts weren't capable. So first, do you have evidence to suggest that your broccoli sprouts weren't adequately inducing NRF2, and then secondarily, the brand new non-canonical method that Donna Zhang showed about a non-KEAP1 mechanism of activating NRF2, which may also be relevant for Tom's newer work, not to Tom's, but are you confident that this is the typical NRF2 where the broccoli sprouts weren't sufficient and the more potent method is that the real mechanism, or is it this new?
Well, that's what I say. I think that the mechanism of protection of the CDDO-IM work that Tom and Bill did, I think that the mechanism goes beyond the ARE NRF2 pathway. I think that contributes to it. It contributes to the lowering of the adducts. But the absence of tumors, I think, goes well beyond just the ARE mechanism. And I have no idea what that might be, but I think that's an area of a lot of interest because there may be lots of things. And it could be downstream signaling even through NRF2, but not just the GST pathway that contributes to that.
Yeah,
exactly.
Absolutely. I'm sure this will probably
Thanks.
elicit some questions later on when we have the roundtable.
Thanks.
So thank you very much, Dave. Turn it back, I guess, to our